Okay, folks. So we've talked about mutagenic. That's questions came, and this slide should have been done before the questions. Okay, so chemicals that bind to DNA are mutagenic agents, and they could be included in cigarette smoke, benzene, nitrous acid, tar. Viruses can also be mutagenic agents because they insert their own genetic material. Okay, so what is cancer this morning, folks? Question for you. Talk to your shoulder partner. You can't do that. Talk to the person next to you, and then talk to me. What is cancer? What do we understand by that? And the main point is if we're talking about humans, they are human cells dividing uncontrollably to form a tumor which invades surrounding tissues. We think these are caused by a chain. So all of that DNA work you've just done on changes to DNA sequence, we think that causes cancer when there's a change to a DNA sequence of specific genes. Okay, so I'm just going to give you a bit of background. I'll give you full disclosure on this one. My mother died of cancer, and so did my maternal grandfather. So I need to hope that I've got my father's genes and not my mother's for that one. But let's give you some more. I'm not alone. Okay, so 1 in 3 people, this is not on the syllabus, this is just for sake of point. 1 in 3 people will develop it. 1 in 5 will die from it. 2007, 297,000 people were diagnosed with it. There's 200 different types, different causes, symptoms, and treatments. And an individual's risk of developing into cancer depends on age, lifestyle, and genetics. And so there are quite a lot of old people get cancer, but obviously younger people can as well. Genetics, if you have, I mean, the great mystery about smoking was not that people died of lung cancer. The great mystery is that some people smoked 20 a day and survived till 80 years old. How the heck was that possible? To which your answer would be strong genes, good genes, we might call them strong genes. Okay, because to continually put carcinogenic chemicals into your lungs, I hope none of you, I wouldn't put any of you down as smokers, but you never know. Anyone here likes a quick fag? Well, no one's going to admit that. No one's going to admit. Liam, are you partial to a quick fag every now and then?
I have 40 a day, sir. Every day.
I thought as much. The big danger for uni students is smoking weed and then getting addicted to tobacco. You cannot put carcinogenic chemicals into something as sensitive as your lungs. It's madness. But if you have very strong genes, it seems that it's survivable. So there are genes there that actually suppress cancer. And you need to ask yourself, do you have them? I mean, I'm sure one day there'll be tests for this. So the smoking is a lifestyle thing. There's living near heavy traffic, isn't it? With the current benzenes are pretty cancer causing. If you're continually living near a polluted roadway, that might be another, not a lifestyle factor, an environmental factor. there's probably a link with weight, obesity, and cancer, but I'm not sure if it's causal or just.
So obesity is like the second highest non-genetic cause of cancer.
Right. I can't remember the mechanism though. Why does obesity cause cancer? I can't. I can't recall, and it's not on your spec to know. I can't recall why would that. I mean, there's loads of things. I can see why obesity would cause pressure on the heart because, if you have a lot of fat in your stomach, your lungs can't expand fully, so you can't get breath easily. And so there's a strain on the heart, plus there's fat in your arteries that would block the flow and lead to high blood pressure from the heart. So I can see the connection between heart disease and diabetes, but why should it cause cancer? I can't recall. I'll have to look that up and get back to you. Well, age is kind of obvious. If we're sat in this environment where we receive cosmic rays and sometimes toxic chemicals, so most of these gamma rays from background go straight through our cells and don't hit our nucleus and don't hit our DNA and don't cause cancer. But if you sit in this slightly toxic environment for long enough, you've got a higher chance that one of those gamma rays will hit that DNA, affect those bases, or one of those toxic chemicals will react with those bases in the DNA. It's a straight probability situation, isn't it? The longer you sit in this world, the more you encounter chemicals. Now, I've put this up here. I think this is just interesting in the thinking about COVID-19, where we've seen 40,000 deaths. If COVID so far, we've seen 60,000 excess deaths, 60,000 excess more than normal in the UK. Some of those could have been cancers that didn't get treated because of COVID. We normally see about 40,000 from lung cancer in a year. And we're talking since February, we've had 40,000. So we are, I don't want you to think that the lockdown has not been a good idea. I thought at first it was a bit extreme. But seeing looking at this death rate, this is one of the biggest killers, and it's already done that in 3 months. Never mind 12 months. Okay, so this thing has turned out to be a big killer. You could say, well, we need to ban smoking ASAP because about 90% of these will be smoking. Probably we do, but I don't know. That moves across slowly. You'll see breast cancer is a big one, and so ladies will get screened on it. You will have to go for your regular screening for that. Men are bad about going screen for prostate. Isn't that the one that they have to stick something up your bum for that one? I don't know. I'm not a medical man, to be honest. I've never been tested for that. But it's quite a big killer there. Say I'm like most men, I'm not prone to go to the doctor very often, if ever. In some countries, they make you go every year for these kind of tests. Maybe that's the way forward. But always get the tests, I would say. Okay, so let's just put a bit of perspective on what you've been through this time. I think, has, in hindsight, seems to have been justified. So let's look at the difference between germline mutation and somatic mutation. So germline mutation is something that's happened in your to your cells in your testes or ovaries. Okay, and so they are mutations that are passed on to the next generation, whereas somatic mutation could be in any cell in the body. Okay, and will cause cancer in that cell, but it's not something hereditary that will pass on. So I said, you need good and strong genes. So what do we mean by this? Well, so some genes for cancer are passed on, okay? And obviously it's a much smaller percentage, but then there's tons more body cells than there are sperm and egg cells, for starters. So there's a lot that can go wrong in all kinds of different areas of the body. Is it the skin? Is it the lungs? Is it, yeah. So whereas this one, there's only a small area that can be picked up, which is in your testes or ovaries. Okay. So there are some clear hereditary problems. So there's one is ladies that have the BRCA1 and BRCA2 gene, and that makes you 70% more likely to get breast cancer. Okay. I have no idea how you find out if you've got that gene. Okay. I did have a student whose family, I think they probably test you when you get breast cancer, because I taught a student whose mother, that's it, they test you if you've got breast cancer for this gene, because it turned out the mother had it and then the daughter had it. And so in that situation, you've really got to avoid estrogen or keep estrogen levels low. It's a risk factor. So that can sometimes be in the pill or in something called HRT. HRT is hormone replacement therapy, and that's what ladies who are going through the menopause were prescribed and are still prescribed if they're finding going through the menopause really tough and difficult and, I don't know, depressing or something. They can be prescribed estrogen. Okay, so other genes we have, as you're going to see, is tumor suppressor genes. Okay, so how good are our tumor suppressor genes? Are they dominant or recessive? Sorry, are we homozygous or heterozygous for these genes? Okay, if we're homozygous, we're going to be less chance of an environmental issue affecting we've got a backup copy, aren't we, if we're homozygous? So that's what I'm talking about when I say, well, how strong are our genes? And that's what we're talking about when we say somatic changes, environmental changes are often required to interact with the genes that we've inherited. Okay, so this is now year 13 work. Getting near the end here. We've got, might not do this justice, but I'll keep talking. We've got tumor suppressor genes and proto-oncogenes. More about that in a second. So the Sanger Institute has, in Cambridge, have sequenced, DNA sequencing, 529 cancer genes. This seems alarming, but you'll see what it means in a minute. By investigating 115,000 tumors, and they found 20,981 mutations. So I'll come back to that in a minute, what that means, because that sounds very alarming, but may not need to be. Oh, sorry, now it's gone up to they found 90,000 mutations, and I'll show you what that means. So first of all, this is on the syllabus. So if you have these genes, they're a control mechanism to prevent out of control cell division. Okay. Now, if this gene becomes mutated, which on your syllabus you're going to say often comes about through methylation or hypermethylation, is what you're going to use in your answer to these questions. Tumors are not going to be suppressed, okay? So that there's different tumor suppressor proteins, okay? They can repress the cell cycle, inhibiting cell division, okay? They can link the cell cycle to DNA damage. If there is damage to the cell, these genes will not allow the cell to divide. If they see a very damaged DNA, these genes will actually do something called apoptosis. Apoptosis this morning. So apoptosis is can also be called cell suicide. Okay, so these genes, quite a risky proposal, but they will notice that the cell is becoming cancerous, and they will put in place a sequence, a complex sequence that you don't need to know about, that results in cell suicide, which you're going to call by its proper name, apoptosis. And so this is a strong preventer of cancer in someone. So it's a strong genetic preventer of cancer. Now, this is a gene with two alleles, okay? And this gene will still function. It's like a dominant gene, so it will still function if there's only one copy, okay? So if one copy gets damaged by the environment, or you are heterozygous for this gene, the other copy will work fine, okay? So that's a sort of a dominant gene. You only need one allele, and your tumor suppressing abilities will still work very well. Thank you. Okay, so that is, this diagram is showing the cell cycle, and here we're seeing a prevention of the cell cycle, which, by the way, is how a lot of cancer drugs work. We try and inhibit the cell cycle. We inhibit mitosis. This is why you need to learn about mitosis, folks. We need to be able to stop mitosis, to stop cancer. A proto-oncogene is something that will normally stimulate division, okay, which is sometimes a good thing, but can be a bad thing. So normally they promote cell division. They release proteins that promote cell division in a controlled manner, okay? And that might be necessary. I don't know, for example, if you hit puberty, you desperately want to grow tall, you need proto-oncogenes that will promote cell division, and you grow nice and tall, those of you who want to grow nice and tall, okay? However, when mutated by hypermethylation, they become known as oncogenes. Oncology, study of cancer, okay? Become known as cancer. And the downside of this one is that you tend to need, this is more like recessive, you need both genes correct for this to properly function. So any sort of mistake on one of those, and we can have fast cell division. If you have an oncogene working, you have fast cell division. So this really affects the rate. And if you can't turn this off, the rate of cell division just continues and never slows down or stops. So the way this cell cycle is turned off and turned on is this protein made by the protein oncogene being produced. Yes, then it will turn the cell cycle on. Is this not producing the protein? Is this switched off at this point? That will turn off the cell cycle. The rate goes down, folks. Okay, so that's it. That's all we need to know about that. We do need to remember the term. This is hypermethylation, is how the mutation is occurred. So that's just adding a CH3 group to a is it to the gene or the protein? I can't remember. I think it's to the gene. Okay, to the DNA. Okay, so as said before, I just must deal with this idea. There's over 90,000 mutations now been discovered in 13,000 genes. How has this been done? Well, we've sequenced the DNA in tumors. So tumors, as I said, I should say a couple of things about tumors. They can be benign or malevolent. Okay, so benign is weird that you can have a tumor growing in your body and it's not cancerous. And it can actually be there for a number of years. So it's just a lump. It's a lump, folks. It's a lump of cells. Self-contained, perhaps growing at a quite slow rate compared to other cancers. It's a lump in your body. And it turns out, for some reason, the rate of growth is slow enough that it's not, or whatever the reason, it's not all lumps in your body cause cancer. Some lumps, however, turn cancerous, and that's particularly when they start disrupting other cells around and disrupting the function of other cells and tissues. And then the worst of all is when some of these get into the bloodstream. Then you know you're in trouble, because these cancer cells will end up being pumped all over the body, and they can just hop out of the blood and start dividing in this tissue and that tissue, and next thing you're just riddled with cancer all over your body, and you've had it, basically. Although I can't think, there is a new treatment on blood. I can't remember what it was, blood cancers. So these tumours, the Sanger Institute at Cambridge have started sequencing all the DNA in them. Okay, and so what they found is, I'm going to skip all of that. They compared the genes in the tumor here with the genes in the DNA. And so this will be done by a technique that you'll learn about in year 13 called gel electrophoresis. We work out the different bases. You can see there's a key here, C, G, T, and A. And we can read off, so for example, that one clearly is C. Okay, we read it off. And for the same piece of DNA, we read off both. In fact, that's probably, I think normally at this point, I would give you a worksheet and ask you to crack the codes there. But I can't seem to give you a worksheet these days, so it's a bit of a pain. So I'm just going to do it for you. Here's the healthy DNA. Here's the tumor. So it's very, very similar in many ways. You have to study it in quite a bit of detail. So each, thinking back, 90,000 mutations. What does that mean? We're talking 90,000 subtle differences in the tumor compared to the original cell. Remember, the DNA in each cell is the same, actually, no matter what cell. But the tumor cell has a whole load of different mutations. So the theory is then that mutagenic, either mitosis or mutagenic elements in the environment, have caused a change to these bases, okay? And that has led to, it's led to the tumor itself. So it's quite, again, really you need a worksheet to spot where's the blooming difference in this code. So despite it being sort of thousands of differences, that's because they've found thousands of different types of tumors. Sorry, how many different tumors have they found? 370,000 different tumors. That's because, you know, there's a tumor in your skin, there's a tumor in your ovaries, there's lots of different parts of the body where they can find a tumor, okay? And you can get lumps all over the place, folks. So That's what it means. There's a lot of different tumours around there, and in each different tumour, there's a lot of different differences in the base. So we think this is the key, that these different bases are the key. Just trying to spot, there should be a difference, but I'll be damned. Yes, I think it might be easier to look at the graph. I think the difference appears to be, look, we've got GTT here and GGT here. And if you look, that's the only mutation there. Okay, and so that is one of 90. That is a mutation. There it is. It's one base difference. It's not a frame shift, so it might not be that serious in that case. But we have found 90,000 to date different variances here between tumors and normal cells in the DNA. Okay, and so we think that is part of the problem that leads to uncontrollable cell division. Oh yeah, there we go. It's identified it. And so typically, this is how they work. It's typically one base is different on these tumors, and there's a one amino acid change to the protein. Okay, and so here's the protein. So this is a protein that's very much linked to cancer. Remind myself what it does. I can't recall. This is slightly beyond what you need to know. I'm just trying to give you. Okay, this is beyond what you need to know. So all I want to tell you about CRAS is it's cell signaling. Okay, so it's the way one cell communicates with another cell. Okay, and if the communication goes wrong, we get inappropriate cell growth, and many cancers have mutated CRAS. So it's a really good place to look for problems, okay, and diagnosis. So what is it? Well, it's just different mutations will have different impacts on the CRAS protein. So remember, this is a signaling between cells, telling them presumably what to do, whether to divide or not divide. Okay, so there's a mutation that actually led to a change in this amino acid. The whole shape of the protein looks fairly similar at the moment, but there might be some binding issues there. So this is very normal in cancer to find slightly weird proteins, slightly different proteins in this area, slightly different proteins in this area. Okay, so that's what I'm trying to get you to understand, how mutations lead to differences in amino acid coding, which leads to difference in shape of protein, which can lead to cancer.